Hi, my name is Alejandra Rubinstein. I'm a food engineer and a PhD student in chemical biology. I work in the Laboratorio Interdisciplinario de Dinámica Celular y Nanoherramientas y Cupin en CONICET, which belongs to the Departamento de Química Biológica de la Facultad de Ciencias Exactas y Naturales de la Universidad de Buenos Aires, Argentina. Today, I'm presenting structural analysis and modeling of the Cupin 1 domain present in the quinoa 11S globulin together with Laura Lucas and Oscar Perez. Quinoa, Chenopodium quinoa wild, is an ancestral crop of the Andean people, which has resurfaced in the recent years due to its nutritional properties and its resistance to abiotic stress. The seeds of this pseudo-cereal contain a high amount of proteins and an excellent balance of amino acids, which exceeds the standard of the Federal Agricultural Organization. One of the main preserved proteins is the 11S globulin, whose structure is currently not crystallized in the Protein Data Bank or PDB platform. Not only this quinoa protein is not on PDB, but there is not a single quinoa protein with its structure crystallized. The objective of this work was to determine the 11S quinoa proteins structural alignments by homology and conserved regions, domain, and motif that were similar to other proteins. Using BLAST platform against RESP-seq, the best hits were proteins that haven't got their structure crystallized. Then we performed BLAST against PDB, establishing Blossom 62 as matrix parameter with a gap cost of 11, extension 1, and word safe of 6. The best hit in this case were the crystallized structure of 11S proteins present in Bertholletia excelsa, Amaranthus hypochondriacus, the same botanical family of the quinoa plant, Pisum sativum, Prunus dulcis, and Cocos nucifera, with a maximum score of 43.85 and an e-value of 1 times 10 to the negative 115 power. In the table, you can see the query cover score and the e-value for each one. It's important to highlight that in the 5 homologates, the query cover was more than 80%, which indicates that the homology is in the whole protein molecule and not only in a region. Once we found the best homologate sequences that we had their structure crystallized, we searched these proteins along with the quinoa 11S in PFAM. As you know, PFAM is a secondary database that groups protein according to the conserved regions. In all the proteins that we searched in PFAM database, the Cupin 1 domain was the common motif with an average bit score value of 104.3. and an average e-value of 4.5 times 10 to the negative 26 power. The Cupin 1 domain is a diverse superfamily of proteins containing a conserved barrel domain and includes both enzymes and not enzymatic sick proteins. The characteristic Cupin domain comprises 2 conserved motifs, each corresponding to 2 beta strands, separated by a less conserved region composed of another 2 beta strands with an intervening loop of variable length, as you can see in the figure. Using cluster omega and Shalview, a multiple alignment was performed, which allowed to determine the most conserved amino acid in the Cupin domain remains conserved in all the proteins. Finally, from DMG, it was possible to visualize the secondary structure of the Cupin 1 domain, as you can see in this last image. From all the analysis carried out, it was possible to say that the homologs found can be used to model the quinoa 11S protein by homology. In conclusion, the bioinformatics result undoubtedly contribute to this domain characterization, which is relevant for the prediction and modeling of the quinoa 11S protein for other practical and technological applications, for example, protein and bioactive compounds carrier designs. Thank you.